Hi everyone, let's begin with the next chapter, and that is culture media. Now ideally, if you're a second university exam student, you would be reading this in the beginning of the course because you will consider it as a part of your general microbiology because you have your university exams coming up. So that time, this chapter is one of the most difficult chapters to comprehend. It's very difficult to understand this because it's way too much of cramming up. But if you're a NEET PG aspirant, I would advise that skip this chapter right now, study the entire bacteriology, and then come back to this chapter, because then this acts as a golden chapter for a summarizing ability. Or if you want to read it, you read it right now, but do revisit it once again, because in the end you will realize that wow, all the culture media have been put into one place. It makes your revision process simpler. Yes, let's begin with the different culture media on that note. First, you need to know the history behind it. So in the history, you should be knowing that there was a liquid culture media, and I think I've taught you in your scientists wala chapter that liquid culture media was given by Sir Louis Pasteur. And then later on came a solid culture media, which was given by the next scientist, that is Sir Robert Koch. So they told you that initially Sir Louis Pasteur, L for L, told us that liquid media, urine and meat broth. Broth is like you have chicken soup broth, like that. So broth or urine, these are all liquidy things. They said that they can allow a lot of bacteria to grow. So they are culture media. Then Sir Robert Koch said that no, liquid maintenance is difficult. So he came up with a solid culture media made up of potato. That is just a historical aspect, because now what we use all throughout universally is agar. So these are 5 questions on agar which you need to know before you begin. What is its origin? Guys, this is going to be the origin from the seaweeds. What seaweeds? So there is a very important family which is the Gelidium and Gracilaria. If you look it up over Google, you will find there are red algae. There are red algae of Gelidium and Gracilaria. So if simply pucha jaye to red algae seaweeds, these are the origin. If someone asks you the composition, it is primarily made up of polysaccharide. It is primarily made up of polysaccharide. What is the nutritive value? This has been a previous year question, guys. The nutritive value is none. None is the answer. It does not have any nutritive value. If you want to add, you will have to add some blood or some serum or some egg. Then it will become nutritious. What is the temperature? So what is the melting temperature? It melts usually above 98 degrees centigrade, and it is then going to solidify. It is going to solidify roughly below 42 degrees centigrade. So we say that half yaad kar lo, 150 ऐसा याद कर लो. So it is melting at 100, solidifying below 50. So 98 and 48, that's the melting and solidifying temperature. What is the concentration? So please note, if you are going in for a solid agar, we are usually using agar, again a PYQ. We are using agar in a concentration of a 2%. However, there will be some situations when you will have to make it a little softer, means you will have to make it semi-solid. Then you will use 0.5% agar. And there will be some cases in which you'll have to make it very firm. So firm for 5. Firm for 5, so you'll have to use a 5 to 6% agar. So obviously, if you've read your bacteriology portion, you would be knowing that when am I interested to make it a little loose? When am I interested in doing semi-solid? See, tell me, if I keep you in a very comfortable, friendly, like if, for example, if I say that, I am your teacher, but I am also a friend, I am also a mentor for all of you. So you'll feel a little more comfortable, you'll share a lot of things, you'll move around, you'll feel open. So then in that case, I have become a semi-solid agar because I'm allowing you to open up, I'm allowing you to share your problems. So basically, whenever you want the bacteria to move, whenever you want to do motility testing, the bacteria will say, I will move whenever the situation is a little loose. Semi-solid agar is what you use. On the other hand, if I'm very like one of those strict teachers who say, if you come late, I will not let you inside class. If I'm very firm, if I'm very strict, you will also be sitting like this, as sit straight back up karke. So then you will feel very rigid. You will feel very closed. So this means if I end up becoming a firm agar, you will not move. If I become strict, you will not move. And that means whenever you want to inhibit the motility, whenever you want to inhibit which motility? Whenever you want to inhibit the swarming motility, who shows you that? Proteus. Then you use a firm agar. So remember, if you use a little bit loose wala, so the organism is going to roam around motile for motility testing. If you make it very firm using 5 or 6%, it will inhibit the motility, both of which are previous year questions. Now, so 5 questions done. It's from the seaweeds made of polysaccharides, no nutritive value, solid, semi-solid, firm. All of them are done. Now let's see what all does agar contain. So agar mein do cheezein to, the agar that we made, now I'm not talking about the agar that you get from algae, what do you make in that culture plate? You use peptone, you add agar, then of course to mix everything you need water and electrolytes. You will also add some different things. Just I'm telling you as an overview, kya add kar sakte? You can add blood, you can add serum, you can add some meat extract, some yeast extract. These in different compositions I will add, and I will get different culture media. So peptone and agar, you will find peptone, agar, water and electrolytes will remain constant everywhere. Then I'll keep playing around with these things and I'll keep making different things. So let me start. The first set of agar media that you have are simple, basic, basal media. Basic rakho. Like you say that, ma'am, these are the organisms which are non-fastidious, non-fussy. Non-fastidious, I mean non-fussy. They are the ones who say that hum grow kar jayenge. You just give us basic food, water, 2 meals a day, we will survive. Those kind of. They don't want any exotic pizza pasta. They want simple food. They are the non-fussy, non-fastidious organisms. They say we will grow in a simple media called peptone water. You can see a water over here. They will grow in peptone water. So peptone and water and salt. Little, they say just give us peptone, water and salt. So there's 1% peptone with 0.5% salt and mix it in water. So we want simple home cooked food, right? To this peptone and water and salt, if I end up adding just something extra known as a 1% meat extract, that now becomes a nutrient broth. Remember, wherever the word broth has been used, जैसे you must have seen chicken soup broth, meat broth, तो कुछ तो meat add हुआ है. So in nutrient broth, you will say, now let me tell you. Now let me explain to you. When do you call it a nutrient broth? When you've taken the same peptone water, wahi peptone water salt liya, in that you added some little extra. Once a while everyone wants little chicken, non-veg meat. So you add that, you get a nutrient broth. But you don't want it to be a broth. You say, mujhe to agar chahiye. I want a nutrient agar. So what do you have? You have the same peptone water. You had converted it into a broth by making meat. Now to that you say, if I add agar, how much agar? Of course, we always add 2% agar. What will I get now? I will get a nutrient agar, and this is the picture, this translucent kind of yellow thing that you have, that is going to be the nutrient agar. So I'm repeating all of this once again. अब छोटा सा एक summary table बना लेते हैं. If I have peptone water, just peptone water and little bit of salt, that is peptone water. To that, if I add 1% meat extract, that meat extract broth hai, so this will become a nutrient broth. To that, if I will add 2% agar, that will now become a nutrient agar. Your simple meals are done, but ये तो simple, non-fussy people who are okay with little bit of, basic food, home-cooked food are okay. Then there are some people say that no, we are fussy organisms, fastidious organisms, we need some good food, pasta, pizza and all of that. So they say, please add extra nutrition. We want egg, blood, serum, something should be added. So what are the enriched media? The first two are blood agar and chocolate agar. Blood looks like blood, chocolate looks like chocolate. Common sense. So what blood am I talking about? We usually take a 5 to 10% of sheep RBCs. 5 to 10% of sheep RBCs are taken, and this is the culture plate. You can see that first every culture media has to be autoclaved. So you autoclave it, and to what temperature do you cool it? You cool it to a 50%. So this is gonna be a blood agar. So सबसे पहले agar का concentration क्या होगा? This I'm not going to repeat everywhere. अब समझ जाओ, ठीक है? Agar everywhere is gonna be 2%. Until and unless I tell you an exception, everywhere understand कर लेना, ठीक है? Let's make it mutual. Agar हर जगह 2%. I will not repeat everywhere. I will tell you the extra things. तो blood क्या है? Blood is 5 to 10% of sheep RBCs. Autoclave करा, cool it to what temperature? 50 degree. What is this blood agar used for? You'll say, ma'am, so many organisms are going to grow. So I can say, I have made it rich. I have enriched. Enriched media matlab, you have made it rich by adding some blood and some extra pizza pasta wala enriched media bana diya. But at the same time, can I also say, this is a differential media? What is the meaning of a differential media? Means it's telling me the difference between something. Have we heard that blood agar tells me the hemolysis as alpha hemolysis, beta hemolysis, gamma hemolysis? For which organism do we have this classification? Streptococcus. So blood agar is actually telling me the differences. Kuch difference bata raha hai. Alpha hemolysis, beta hemolysis, gamma hemolysis. Anything that will tell me a difference, that becomes a differential media. So I can say that blood agar is not only an enriched media, It is also a differential media. Now I'm going to do a variation. I'm going to call it a chocolate agar. So what do you see? You'll say, ma'am, sab kuch to same lag raha hai. You have the same 5 to 10% of sheep RBCs. You have the same nutrient agar that was autoclaved, but this time it has been cooled to 70 degree. So remember, in blood agar, it was cooled to 50 degree, and in chocolate agar, it is going to be cooled to 70 degree centigrade. है ना? So please note that why are we doing this? What is the advantage of chocolate? Chocolate is going to release a lot of factors. Actually, there's something called factor V and X, which is very commonly known as factor 5 and 10. That is released and that makes the blood more nutritious. Blood agar also had minimal amounts, but now chocolate agar will have too much of factor 5 and 10. It will make it more nutritious for your particular bacteria to grow. So blood agar mein to nutrition tha, but chocolate agar is going to have more and more nutrition. This is sorted. What else? Then we have another enriched media which is known as Loeffler serum slope. The rule of this subject is whenever Loeffler comes, there's one bacteria coming to your mind, that is Corynebacterium diphtheria. The other name for Corynebacterium diphtheria is Kleb-Loeffler's media. And what have I taught you in Corynebacterium diphtheria? That if someone asks you Corynebacterium diphtheria ka enriched media kaun sa hai, you will say 1, 2, 3, 4, 5, 6, 7, 8. And then you will write Loeffler and you will say, oh wow, that is also 1, 2, 3, 4, 5, 6, 7, 8. And how much time does the Corynebacterium take to grow over here? It is going to take 8. It is going to approximately take 8 hours to grow over here. So repeating once again, enriched has 8 alphabets, Loeffler has 8 alphabets, and the time that it's going to take to grow is also 8 hours. That is the mnemonic that we've done. Up next, what I cannot ignore in our country, you will say that, ma'am, ye to sabko pata hai, that is the LJ media, Lowenstein-Jensen media. Of course, you know which organism I'm talking about. That is tuberculosis. Any kind, not just TB, even atypical TB. TB se related kuch, Mycobacteria family se related, Lowenstein-Jensen media. They ask you the composition of Lowenstein-Jensen media, and I've always taught you that it looks like an entire English breakfast. What all do you have in English breakfast? Chalo, let us see the English breakfast, guys. So please note, sabse pehle you have eggs. So you're going to have something with eggs, that is coagulated hen's eggs. On top of that egg, you have sprinkled a little salt. So you have a mineral salt solution. Then eggs ke saath you will have some green vegetables. You will have some green, malachite green vegetables, like ye jo green dikh raha hai, asparagus. So I've learned green asparagus matlab green asparagine. What is the English breakfast? Coagulated hen's eggs, sprinkle exotic breakfast. Okay, so sprinkle a little bit of salt on top of it. Then you're gonna get green veggies like malachite green and asparagine, and that is why now you understand why is the background looking green, because you have added malachite green. Why? Kya milega? By adding, I could have added some blue, pink, yellow, so many colors. Why have I added malachite green? Because malachite green is going to inhibit the other bacteria. It is not only just a show-off wala green, it is also going to inhibit the other bacteria, and that is why it's very, very useful. It's acting as a, an agent to allow TB to grow by suppressing the others. So remember, this is the LJ media for your enriched. So what are the enriched media that we have done, guys? We have done blood agar, chocolate agar, LSS and LJ media. If you've done your sterilization and disinfection chapter, you should be knowing that all the culture media are going to be sterilized by autoclave. Everywhere I'm saying na, I'll give you an example, that there was autoclaving that was done. Pehle bhi I said there was autoclaving that was done. Sterilized bhi to karna hai. But suddenly when I came to LSS and LJ, in case of LSS and LJ, I said, nahi, I didn't use these words autoclave and, all these things. Why? Because for these, you do something known as inspissation. Go to your sterilization disinfection chapter. You know we've done this over here. That in inspissation, we are going to use when we are dealing with LL. LJ and Loeffler serum slope. We do not autoclave them because in dono mein there is, see, Loeffler serum slope has serum and LJ media has egg. And autoclave happens at 121 degrees centigrade. Very high temperature. Your serum is going to get damaged. The egg is going to get coagulated. So 121 pe to nahi kar sakte. We are going to do it as inspissation, which is a less than 100 degrees centigrade or method of sterilization. So you have to keep the heat low. You've understood the cause. Okay, enriched media done. Now we go to enrichment broth. Now what is this? Ma'am, broth word you have been telling us from the beginning means something that is liquid. Is enrichment and enriched the same? No. Micro hai na, one alphabet changes the story. So now let me tell you the difference between these three things. I know before I start, it's important that I completely tell you the concept behind it. एक होता है enriched media, then there is something known as an enrichment broth, and then there is known as a selective media, right? I'm assuming that all of you always have a confusion between them. See, first make a rule that wherever the word media will be used, you will always deal with something that is solid. As soon as the word broth will be used, you will have something that will be meat broth, chicken soup broth, तो कुछ liquid होगा. तो the consistency is clear. Enriched media means I am providing something extra. Means you are providing extra rich, extra nutrition. You are adding something that will allow you, adding that pizza pasta so that the bacteria becomes happy and grows more. Selective means that you are adding something which will not let the others to grow. For example, selective media is something like Chatur from the movie Three Idiots. That basically means that he has studied himself, he has worked for himself. Now before the exam, he will go and stop others from studying, right? So this is gonna inhibit the other organisms from growing. It will only allow the growth of self. It will not let the other organisms to grow. And what is enrichment broth? You will say, ma'am, broth तो समझ आ गया, liquid है, but enrichment मतलब कैसे? Is it gonna act like enriched or is it gonna act like selective? Let me tell you, it is gonna actually act like selective. It is a liquid media, but that has properties of a selective. मतलब you can say selective media solid था and उसका liquid cousin sister is enrichment broth. Simple. So enriched media solid and you've added extra pizza pasta, extra growth nutrients. These two are selective, means like Chatur from the movie Three Idiots, अपना growth कर लिया but not allowing the others to grow. So broth is gonna be liquid and media is gonna be solid. So they are cousin brothers, they are solid liquid cousin brothers, but their work is the same. Now coming back, I'm talking about an enrichment broth, and the first one that we have is APW, alkaline peptone water. Water के साथ Vibrio is matching. So alkaline peptone water and Vibrio is matching. Let me tell you, in a while I will tell you alkaline peptone water is also used sometimes as a transport media. Okay, let's move forward. We have some broths. Again, the word broth is coming. So you realize na, alkaline peptone water, it was a liquid, तो broth में जाएगा. Selenite F broth and tetrathionate broth is also a broth, so that will go in the enrichment broth. And which two? The SS sisters, Salmonella Shigella sisters require selenite F broth and tetrathionate broth. Everyone's okay? So alkaline peptone water for Vibrio, selenite F and tetrathionate broth for SS sisters, Salmonella Shigella sisters. Now I come to the solid media for this, that is selective media. Like I said, there is a component which will inhibit the other bacteria. That is the story that I was telling you. So the first one is the favorite one, McConkey. McConkey का सबसे पहले I will tell you a composition, then life will become easy. McConkey, if you are asked that what is the composition of the McConkey agar, it is made up of plant. It is made up of plant. Plant means it is P for peptone. Of course, ma'am, every agar has peptone. Peptone and agar to has to go everywhere. Then it is made up of lactose. Why is it made up of lactose? Because guys, this is also a differential media. This is telling me the difference whether the colonies are lactose fermenters or not. So जो difference बताएगा वो differential media. Next we say that this also has something called neutral red. So you must have learned. You'll say, ma'am, how will you get to know lactose has been fermented or not? So look at here. If lactose is fermented or if lactose is not fermented, I get to know because fermentation of lactose will give a pink color, and if no fermentation occurs, it will give a pale color. So color change ho raha hai. That means that neutral red is going to be an indicator for the color change. Can I say this is also an indicator medium? So many functions it has. Lastly, it has something called taurocolate. What is taurocolate? Like sodium taurocolate and all. And taurocolate bile salts, na? So you will say, ma'am, bile salts to bahut toxic hote hain. This means this is that Chatur from the movie Three Idiots who is not letting the other organisms to grow. So this is what is gonna make this media selective, and here you go, this is also a selective medium. Let's do a repeat. What is McConkey agar? It is a selective media. Why? Because of taurocolate. It is a differential media. Why? Because it tells me difference between lactose fermentation or not. And how is that told? By an indicator called neutral red. The color changes. Color change hoga, so I will get what color? I will get a pink color. Let's move on. So this was the first selective media that you studied. The second selective media that you're going to have is going to be the very, very famous mannitol salt agar. So abhi previous wale mein you studied about what? You studied about the McConkey. So the next selective media that we have to know is CLED, because it's on the same lines. I just spoke about lactose fermentation. I'm going to talk about lactose fermentation next, and that is the cysteine lactose electrolyte deficient. So what is this, guys? First and foremost, it has cysteine, it has lactose. Lactose means this is definitely going to tell you about lactose fermentation or not. So is this also going to be a differential media? Yes, by now you've understood the story. Next, it is also going to be a selective media, like I'm telling you, means it is going to allow some organisms to grow and it will not allow the others to grow. It is a selective media. But now you'll say that if lactose is getting fermented, ma'am, what is the color that you get? For lactose fermentation, I can see that I'm getting a yellow color colonies. I'm getting yellow color colonies over here when lactose is getting fermented. But color change matlab indicator. So what is the indicator that has been used? Look at the rest of the background. It's blue. So the indicator used is bromothymol blue. So now when McConkey was also the same, matlab it was selective and differential and indicator. This is also the same. It is selective, it is differential, as well as it is an indicator media. So and दोनों का काम भी same है. This is also telling me lactose fermentation. McConkey was also telling me lactose fermentation. Which is the better one? So please remember, CLED is better than McConkey. Why is CLED considered better than McConkey? Because both of them are like chatur. Both of them are selective. Both of them will not allow the others to grow. But CLED is less inhibitory, or you can say mildly selective. Mildly it is like chatur, very little traits it has, and that is good, because please note, for example, you have a case of urinary tract infection and you've taken a urine sample. A urinary tract infection can be because of gram-negative organisms like E. coli, and it can be because of gram-positive organisms also like Staphylococcus and Candida. So you will say that, ma'am, CLED will allow everyone to grow. CLED will allow both gram-positive as well as gram-negative organisms to grow. However, McConkey will only and only allow the gram-negative, the usual wala, to grow. It doesn't allow the gram-positive that much. So that is why I say that CLED is better. It is more accommodating. Okay. So two media are done under selective. Let's move on to the third one. The third one is now lactose bahut ho gaya. Let us talk about mannitol salt agar, mannitol fermentation. So again, the same story. This is also selective and differential and indicator. I'll tell you how. Firstly, mannitol salt agar has salt in it. Salt makes it selective because salt will only allow Staphylococcus aureus to grow. It does not allow all the others like Micrococcus and other cocci to grow. So selectively only one thing is growing. Then so salt matlab selective. What is mannitol? I am wanting to know whether mannitol has been fermented or not, because I know that Staphylococcus aureus is going to be a mannitol fermentation, whereas pick up any other Staphylococcus like Staph epidermidis, and that is going to be a non-mannitol fermenter. That is not going to ferment mannitol. So difference pata lag gaya, it is a differential. Salt told me it is selective. Mannitol is telling me it is a differential media, but if color is changing for mannitol fermentation, what color did you notice? In mannitol fermentation, I've seen a yellow color over here. So this means color change hua. It has an indicator. What is the indicator? It is phenol red. So it is an indicator media also. This was for mannitol fermentation, Staph aureus. The last fermenter that I'm coming to is TCBS. Now what is TCBS? It is going to be thiosulfate citrate bile salt sucrose. Thiosulfate citrate bile salt sucrose. So now this is going to tell me, okay, again wahi story. Firstly, it has bile salts. Does that make it selective? Yes, bile salts are very chatur, toxic. Sucrose, this means now I'll get to know about sucrose fermentation being there or not. So this is also telling me a difference. This is also a differential media. Chalo dekhen. So if this is Vibrio cholera, this is a case of Vibrio cholera, and this is a case of Vibrio parahemolyticus. This is Vibrio cholera and this is Vibrio parahemolyticus. Vibrio cholera is a sucrose fermenter. Vibrio parahemolyticus is not. So when sucrose got fermented, what color did you notice? You noticed a yellow color. You noticed a yellow color. Otherwise, it's all green. So again, if there is a change in the color, you will now again ask me the indicator. The indicator is BTB, that is again bromothymol blue. So this means this is also an indicator media. Matlab ye jitne bhi fermenters you taught me, ma'am, lactose fermentation ke McConkey and CLED, Mannitol fermentation का mannitol salt agar, and this TCBS for sucrose fermentation. All of them are differential selective indicator. All of them are going to be DIS, that is differential selective and indicator. So now you are going to make a final list for me so that we cannot have any confusion. And I know you're having a confusion, kiska kya indicator tha, what was the use, what was the color. So never mind. So first what we'll start, we have three things in hand. So let me first start with mannitol fermentation. I'm saying it is a mannitol salt agar. Can you quickly tell me which is the main organism for which you're going to use it? You're going to use it. Mannitol fermentation is shown by Staphylococcus aureus. So now you need to just tell me two things. Color change, kaun sa indicator did you use and what color did you get? So in mannitol salt agar, the indicator was phenol red, and if mannitol was fermented, the color that we got was yellow. Next, I have sucrose fermentation, thiosulfate citrate, bile salt, sucrose. Who is going to show you sucrose fermentation? The organism is Vibrio cholera. So now you will ask me what is the indicator? The indicator was bromothymol blue, and when sucrose was fermented, you were getting a yellow color. You will actually realize everywhere it's yellow. Now let's come to the lactose fermentation. You had a media called the Cled media. Cled media is going to be for lactose fermenters, right? And then you had another for lactose, that was MacConkey media, that was also for lactose fermenters. So now tell me, both of them are going to do lactose fermentation, and Cled also gives you a yellow color for lactose fermentation, but the indicator was bromothymol blue again. Whereas MacConkey ka to wo mnemonic tha na, plant had taught you that neutral red was the indicator, and finally you should know that here the lactose fermentation is going to give you pink. So I have learned it is a pretty pink color plant, because now you will realize that everywhere you get a yellow color. Any fermentation happening on this planet, you are getting a yellow color. Chahe wo mannitol fermenter ho, ya sucrose fermenter ho, ya lactose fermenter ho, fermentation matlab yellow color. Except for MacConkey, which is a pretty pink plant where the fermentation is going to give you a pink color. This column is simple. All of them are yellow. Only MacConkey is pretty pink plant. And what are the indicators? So MacConkey ka to plant neutral red, you will know. Mannitol salt agar is phenol red, and the others are all going to be bromothymol blue. That is the most golden slide you have, because these are differential and indicator and selective media that you had to know. Let's move forward. Now the selective media are not over. Ye to sirf fermenters the. Now you need to quickly answer these now. Where do you use crystal violet blood agar? So we had learned that the way you write Streptococcus pyogenes, हमने pyogenes का हमेशा V बनाया था. So we had said crystal violet blood agar is going to be for Streptococcus pyogenes. Modified Thea-Martin medium is for what? It is for Neisseria meningitidis and Neisseria gonorrhoeae. Both of them have the modified Thea-Martin medium. Cetrimide agar, if you remember, we had a mnemonic for Pseudomonas. We had said that Pseudomonas is gonna be the Pseudomonas burns patients में होता है ना, तो Cetrimide burns में. Remember, Pseudomonas is for Cetrimide media. Plet was going to be the very famous Bacillus anthrax. So Bacillus anthracis का selective media. And MIPA, that is mannitol egg yolk phenol red polymyxin. MIPA के साथ एक उसका cousin brother भी था called PEMBA. MIPA and PEMBA were what? So if you remember, this is the third time I think in this entire syllabus I'm giving you this mnemonic. There's a very famous speech of a very serious politician, and her speech says, 2 years ago it went viral on social media. Her speech had words like rumba rumba and all these words, if you remember that speech. So rumba rumba always, since that day I have always learned rumba rumba is MIPA PEMBA. And she is a very, very serious politician. तो serious वाला जो organism है, वो serious politician, suddenly in the middle of her speech she started speaking words like rumba rumba and all. So yeah, so MIPA PEMBA reminds me of rumba rumba. So serious वाला politician, Bacillus cereus. If you don't know whom am I referring to, Google is always there for you. Okay, now the story is not over. Anything with deoxycholate citrate agar and xylose lysine deoxycholate, these are for the Salmonella and Shigella sisters. So Salmonella and Shigella are going to be the deoxycholate wala. So Salmonella Shigella sisters will have a lot of commonalities which I will tell you. Abhi tak I have told you the commonality of their broth. So we will keep this slide for our Salmonella Shigella sisters. Selective media you have studied, anything to do with deoxycholate. If I would have asked you कि दोनों का please मुझे enrichment broth बता दो, so you will say for both of them it was selenite F broth and it was tetrathionate broth. For both of them, it is selenite F broth and tetrathionate broth. So both the broths are done. Abhi I'll come back to this in a while. We'll complete the Salmonella Shigella sisters, but selective media khatam nahi hua. Finally, potassium tellurite agar. Anytime the word tellurium comes, tellurium is going to give black color colonies, and this is going to be for Corynebacterium diphtheria. So now we have studied the entire story of Corynebacterium diphtheria also. If they ask me what is the enriched media, remember I told you enriched had 1, 2, 3, 4, 5, 6, 7, 8, and which is the enriched media? It is the Loeffler serum slope, because Loeffler also has 8 alphabets and it is going to take 8 hours. So enriched 8, Loeffler 8, 8 hours. But now if I ask you the selective media, that's in front of you, potassium tellurite agar, and it takes 48 hours. So enriched was 8 hours. Selective takes 48 hours, and potassium tellurite agar is basically gonna be black. Tellurium black. All selective media are done for you. The next one I don't have to teach you because I have already taught you. If I individually ask you what are the differential media, you will say, ma'am, not just these. I know many. I know something called McConkey. McConkey is definitely going to tell me a difference because it tells me difference of lactose fermentation hua ya nahi hua. Similarly, for lactose fermentation, I have Cled media. Lactose fermentation hua ya nahi hua. Blood agar is also a differential media. I had told you why. Because blood agar tells me alpha hemolysis, beta hemolysis, gamma hemolysis difference batayega. After that, I didn't want to repeat, but all the other media that you had, be it a sucrose fermentation, that is TCBS, or be it a mannitol fermentation, that is mannitol salt agar, jitne bhi fermenters the, they were all differential, they were all indicators. They remember they were differential indicator selective dis. Add kya kara? Blood agar, because blood agar also tells me the difference. Now finally I come down to transport media. Transport mein kuch bhi add. See, transport is not that I want the bacteria to grow or I want the bacteria to become very developed. No, transport is media is only to maintain the vitality. It is only to maintain the vitality of the bacteria, that if it has to get transported from 6 a.m. in the morning till 10 a.m, if it has to travel, the sample has to travel for 4 hours, it should remain vital in that process. So Stewart and Amy's media, which are some charcoal containing media, are important for Neisseria. Pike's media, pi for pie. Streptococcus pyogenes. What did I say? Pi for pie. Streptococcus pyogenes. VR and Carrie Blair media, V for V, they are going to be both of them. So VR is Venkatraman Ramakrishnan and Venkatraman Ramakrishnan was for Vibrio. So remember the VR and Carrie Blair media are for Vibrio, and the Sachs buffered glycerol saline, SS sisters. SS sisters. What are these? SS best girls. What are these? Who are the sisters? Salmonella Shigella sisters. Okay, so the Sachs buffered glycerol saline are going to be the Salmonella and Shigella sisters. This means I've added to the story of Salmonella Shigella. I can now quickly write down what is their transport media going to be. Ek aur cheez mil gaya. What is their transport media? The transport media for both is Sachs buffered glycerol saline, the SS. Right? Their broth was selenite F and tetrathionate, and their selective media was DCA and XLD. So all the transport media are done for you. Now the next one, almost towards the end, you say, I don't want any oxygen. I am dealing with anaerobic organisms, and I think you know that family. The family is Clostridium. Clostridium tetani, perfringens, they don't need oxygen. So you can't keep it like, you can't say that, okay, this is a culture plate. Isme to itna zyada oxygen from the surrounding hai. You need a place where all the oxygen has been cut off, hai na? All the oxygen has been cut off, and for that you use something called Robertson's cooked meat broth. I'll tell you some containers also on how it is used. So this is known as Robertson's cooked meat broth. I've understood that broth hai to kuch to it must be some liquidy media, hai na? Isn't it? Broth means liquidy, and it can show two reactions. It can show a proteolytic reaction. It can show a saccharolytic reaction. Proteolytic and saccharolytic. So question comes simple in the paper. Proteolytic and saccharolytic. So which of these is going to be seen in perfringens and which of them is going to be seen in Clostridium tetani? So please remember, perfringens is going to be saccharolytic. Perfringens is going to be saccharolytic, and the color that it's going to get is pink. So how have I learnt it now? By now, I don't know how many of you have seen my path lectures and how many of you have seen the IELTS of microbio, like mycology ka lecture dekha ya nahi dekha. You must have seen that ma'am, while making mnemonics, is obsessed with two lectures. Once she keeps saying PP wala mnemonics she's very fond of. Second, she's very fond of Chatur from Three Idiots. She makes a lot of mnemonics on him. And third, she makes a lot of mnemonics on her own name, because when she can't think of something, she comes down to her own name. Self-obsessed. So what is this? So per and sac, matlab perfringens is sounding like my name Preeti, and sac is sounding like my surname Sharma, and my favorite color, which is quite coincidental, happens to be pink. Okay, so having that, perfringens, which shows you a saccharolytic reaction, is gonna be a pink color reaction. Kya likha hai? Perfringens, which is a saccharolytic reaction, is gonna give the meat a pink color. Ab baki sab ulta. Proteolytic is gonna be seen in tetani and is gonna give a black color. वो अलग करना, ठीक है? तो एक mnemonic याद करो, perfringens, which is saccharolytic, so Preeti Sharma pink is going to give a pink color, ठीक है? But you say that, ma'am, for example, I have this kind of a container. इसे क्या बोलते हैं? There is, I'll come to the pathogenesis, but this is known as the Mackintosh and Fildes jar. You will say that, for example, I have a culture plate and I want to make sure that इस culture plate में कोई oxygen ना हो. What will I keep it inside? I will say, for maintaining anaerobiases, you can keep your culture plates in the Mackintosh and the Fildes anaerobic jar. तो क्या क्या है इस jar में? I can see I've kept all the culture plates like this, right? And I can see there is some catalyst over here. Lid को अच्छे से ऐसा tight कर दिया. Look at this screw लगा के you have tightened it. No oxygen should be there. But जितना भी try करो, some amount of oxygen will be over here. So what do? I will have to make sure that I put something that whatever little traces of oxygen is there, that is also absorbed. So what do? When I keep these culture plates, along with these culture plates, can you see I have kept something like packet over here? Can everyone appreciate this packet? Yes. And what all do you see in this packet? Gas pack bhi naam hota hai, which I'll come to in a while. This packet mein I can see there are some, some airs, like there are some molecules, carbon dioxide, hydrogen, nitrogen, ये सब रखा है पहला. Second, we have also kept something called methylene blue. We have also kept something called methylene blue over here. So now I'll tell you the concept. तो देखो, methylene blue is gonna be blue till the time oxygen is present. जब तक oxygen है, methylene blue will look blue. However, now remember, if I open this, पहले तो this packet was closed. Now I have opened this packet and all of the CO2 and H2 have come out. Will they utilize the oxygen? That is what you wanted to do, na? You wanted to take away, suck out all the oxygen. They have utilized the oxygen. They have made things like water and whatever. The H2 with O2 water bana, chemical formula hua. Basic thing is oxygen has been removed, and as soon as oxygen will be removed, this methylene blue will become colorless. So I will get to know कि ठीक है, if the blue color suddenly over here becomes vanished, it becomes colorless, I'll say okay, oxygen गया तो simple, blue color है तो oxygen है, blue color नहीं है तो oxygen नहीं है. That is what you see over here, that if you have oxygen, it will be blue, and if there is no oxygen, if the colorless, if there is a reduced methylene blue, matlab if the oxygen has been taken away, this means they are going to get a colorless reaction. So simple, blue hai to blue O. I don't know how many of you remember, but at least in Delhi there was a very famous bowling shop called Blue O. So blue hai to matlab oxygen hai, and blue nahi hai to matlab oxygen nahi hai. That is the Mackintosh and Fildes jar. But now we have something which is a little more, it's a little more advanced, and that is known as the gas pack. So you have these kind of containers, okay? You have these kind of boxes, keh lo. These are boxes like this, and inside those boxes, can you see you've kept a lot of those culture plates, and they are going to do the same thing. These are also a method of anaerobiasis. But you'll say, ma'am, isse to it's taking a lot of manual effort. Do you have some anaerobic methods which are, you can say, something to do with the machine also? Yes, kuch machine se hai, kuch aise packets mein se hai, right? So we have something called an exomat. We have a glove box or an anaerobic chamber. We have something called PRAS, pre-reduced anaerobic system, and I just showed you something called gas pack. ये जो container दिख रहा था, which is the method of choice for anaerobiasis. You have a container like this and you can keep the culture plates inside it and maintain anaerobic environment. So this is the latest one that I'm talking about, the gas pack. MCQs के लिए gas pack is important. For second prof students, you have to look into all. So finally, all of your culture media are sorted. Very, very rapidly, 1 minute mein let's flip through all. The first one that we studied was a simple media. Simple was very, very simple, just peptone and water. To that peptone and water, when I was adding 1% meat extract, I got nutrient broth. I added 2% agar, I got nutrient agar. Simple. Enriched, make it a little rich. Blood agar, chocolate agar, which you were autoclaving, hai na? Blood agar was cooled to 50 degree, chocolate agar 70 degree. And what were the other L and L? Loeffler serum slope and LJ media. Loeffler serum slope for Corynebacterium, LJ media for TB. These are all enriched media. Coming to enrichment broth, they had to have some water. So alkaline peptone water, Vibrio. Selenite F broth and tetrathionate broth for the Salmonella Shigella sisters. Now coming to the next one, the selective media. Quickly tell me, McConkey and Cled for what? Lactose fermentation. In the chapter of lactose fermenters, you will study something called eosin methylene blue. That is also a lactose fermentation ke liye. Mannitol salt agar, mannitol fermentation. TCBS, sucrose fermentation. Crystal violet blood agar, Streptococcus pyogenes. Thayer-Martin is going to be Thayer-Martin, that is Neisseria. Cetrimide, Pseudomonas, cetrimide. Plet is going to be for Bacillus anthracis. And Mypa Pemba, I told you that Rumba story for a serious wala politician, Bacillus cereus. Coming to the next, differential. So many of them we've done. Lactose fermenters, sucrose fermenters, mannitol fermenters. Sab difference batayenge. And blood agar also tells me the difference. Transport media, tell me these two. Stewart and Ames is going to be for Neisseria. VR and Carrie Blair is for Vibrio. Pike's media is for Pyogenes. And Sachs buffered glycerol saline is for Salmonella Shigella. Finally, anaerobic media, Robertson's cooked meat broth. Tell me Clostridium perfringens. Clostridium perfringens shows you proteolytic or saccharolytic. Yo Preeti ki tarah tha, so it must be showing me saccharolytic, and favorite color pink, so that is going to get me a pink color. Finally, all of these media are done. Now what I need to tell you are two things. How do you make a culture plate? Pehla. And second, I need to tell all of you the different antibiotic sensitivity modalities. So when I'm talking about the different culture techniques, the first one, so if you have a culture plate, so for example, if this is your culture plate, how are you going to streak through it? How are you going to make the culture plate? So first thing is known as a streak culture. What is a streak culture? You can see this. I've got you a diagrammatic representation of this streak plate isolation method. What do you see here? Ma'am, sabse pehle to you took a loop. You took a loop called the nichrome loop. Okay? You took a loop called the nichrome loop, and in that nichrome loop, pehle to use sterilize karo. So the first thing that you will have to do, usually we take a nichrome loop and we are going to sterilize it. Once you over the flame, you sterilize it, then you take the sample. Urine ka sample hai, blood ka, sputum ka, jo bhi sample hai, you dip it in the sample, and now you are gonna start streaking. So can you see you have made one streak here? Then after that again you repeat the process. Sterilize, pick up the sample, pehle streak kara. Okay, then again sterilize, pick up the sample, 90 degree pe streak kara. Sterilize, pick up the sample, 90 degree pe streak kara. So simple, I'm going to repeat that again. Sterilize, pick up the sample, streak it. Then again sterilize, pick up the sample, streak it at again 90 degrees. Sterilize, pick up the sample, streak it at again 90 degree. Sterilize, pick up the sample, streak it at 90 degree. This is one methodology. Some also you must have seen, do it one followed by second, followed by third, and dot dot dot. So that you get very, very pure colonies and the concentration keeps on decreasing. So many methodologies of streak are known, but the concept is that to obtain pure colonies, bilkul aisa streak you are putting, and that is known as the streak culture. Versus if you do the same thing in a test tube and you again take sterilize, pick up the sample, and you're gonna stroke it out. Like over here, you're gonna put your loop inside and you're gonna stroke it out. So that is done on an agar slope or slant. This is also very important. This is also gonna give you pure test, and jo bhi growth aayega baad mein, that you can use for different type of diagnostic tests. Example, if you want to test for urease, if you want to test for urease, you want to do any slide agglutination test, so jitne bhi testings ke liye hai, you can do the stroke culture. Coming to the third one, there is something called lawn culture. Lawn culture is also known as a carpet culture. lawn culture is also known as a carpet. So like you must have seen in a lawn in a park, or in a lawn if you have a grass carpet, है ना? So please note, like that, if this is your culture plate, just take the sample, like you can see na, cotton bud se kya kara, cotton bud se sample liya and spread it all over like a carpet. You have put the urine sample on the culture plate. You took the urine sample and like a carpet you have put it onto the culture plate, not streaking, not stroking, direct like a carpet ki tarah bichha do. That is known as a lawn, or a, you can say, a kind of a flooding that is there. And the most common usage of this is antibiotic sensitivity testing. Antibiotic sensitivity testing, which I am only teaching you in exactly 5 or 7 minutes. Whenever this is actually a diagram of antibiotic sensitivity testing, over there you use a lawn or a carpet culture. The next one that we have is the gelatin stab. Now please note, till here I was saying sterilize the loop, pick up the sample. What was I saying? Sterilize the loop. Till here you were using a nichrome loop. Now cut out the loop. Now you're going to use a straight wire. Why are you going to use the straight wire? Because अब क्या करना है? Stab करना है. Stab वैसे करना नहीं है, but how is a stab shown in movies? Take a knife, one time stab. You don't stab multiple times. तो वही one go में murder करना है. What have you got? You have got a gelatin culture media, a gelatin agar over here. If you would have taken a loop, अगर loop वाला कुछ लिया होता, you take the sample, if you put it in, you put the loop right till the bottom. As you will take the loop out, don't you think gelatin will also get stuck in this loop and start coming out? So difficulty hoga. So we said don't take a loop. Ignore the loop, take a wire. Take the sample, one time in and out. Khatam. It's gonna be a simple. You take the wire like this, one time in and out. Just one time in and out. You're not supposed to do any multiple stabs, and that is a stab culture. What am I trying to see? I am trying to see gelatin liquefaction. Can you tell me this picture shown over here? I see an inverted fir tree appearance. I see an inverted fir tree appearance. So please note, inverted fir tree appearance on gelatin is shown by Bacillus anthracis. Bacillus anthrax. Whereas a seedha fir tree appearance is going to be shown by the very famous Clostridium tetani. So many people ask me that, okay, these are the two test tubes, and in one of them you will see ulta wala tree, inverted fir tree. In one of them you will see seedha wala tree. So remember, tetani shows you seedha tree, TT, tetani for tree, and Bacillus anthracis, ulta wala tree. For INICET students, let me tell you, where do you see the napiform or the turnip wala pattern? Napiform or turnip is seen with Vibrio cholera. You don't get a picture of it, but this question comes. Here the gelatin liquefaction will look like a turnip, like this. So napiform or turnip liquefaction is seen by Vibrio cholera. How I have taught you in the Vibrio cholera chapter, yaad aa raha hai? I told you Vibrio cholera shows you what stools? Rice water stools. So rice you can't have alone. You need some vegetable also with it. So rice ka combination turnip ke saath kara tha. So rice water stools with turnip liquefaction. So how the gelatin is breaking? Is it breaking in an inverted fir tree manner, in a seedha wala tree manner, or a turnip wala manner? The manner is different. So these are the three questions that you get on gelatin liquefaction. Now you have the next thing, and that is known as two techniques. One is known as a pour plate method. One is a pour plate method, and the other is going to be the spread plate method. So what is the difference between them and what is the utility of them? Pour plate method, spread plate method. You will say, ma'am, whatever method you do, ultimately you have two things. You have agar and you have patient ka sample. So think, I can either put the agar first and then the patient ka sample, or I can put the patient sample first and then the agar. I have no third option. There are only two things, agar and patient sample. What will you put first is gonna make the difference. So in a spread plate method, what do I have? I already have the agar that is present in my tube. In this particular slide, in this culture plate, agar pehle se hai. What have I added over here? I have added the patient sample. Whatever sample you had, basically bacteria, na? Jo bhi bacteria tha patient se, you have added the bacteria. So simple. What have you done in spread plate method? In spread plate method, I have added agar followed by bacteria. Whereas in pour plate method, you will do ulta. You will add the bacteria first. So you actually have the bacteria first, and then on top of it you have added the agar. So that is why I said you will have to make a mnemonic for one slide. So remember, spread plate method mein sub. Spread plate method mein what do you add first? You add the agar first and then you add the bacteria. Whereas in pour plate you add the bacteria first, then you add the agar. So what did you realize? Niche kaun hai? Ma'am, pehle to you had added this bacteria. This blue color bacteria you have added. So niche to actually bacteria hai and the agar you have added on top. So bacteria will grow niche. More or less the bacteria will grow under the surface also and on the surface also. The bacteria growth will be seen on top also and bacteria growth will be seen under also because you added the bacteria niche. And that is what you notice over here. In a pour plate method, bacterial growth is going to be noted underneath also, under the surface also. That is what you have in a pour plate method. So please note, pour plate method is done for quantification. When you have to count, this is the real question. If you ask me the real question for the exam, the pour plate method is done for quantification of the bacteria, because after pouring, you can actually count. You can actually do, obviously no one does it like with the help of naked eyes. These are all machine based things, but you can actually do the quantification of the number of bacteria. So remember, out of pour plate and spread plate, kya yaad kara? Spread plate mein agar and bacteria. Spread plate, first agar, then the bacteria. Pour plate mein ulta. And pour plate is done for counting. That is the quantification of the bacteria. So simple, one word. First you start streaking at 90 degree, that is streak culture. If you stroke it in a test tube, it is stroke culture. If you're going to spread it like a lawn or carpet culture. If you're trying to see the gelatin breakdown or liquefaction, stabbed culture, stab the gelatin. If you're trying to put a pouring method, either pour plate or spread plate, pour plate is something which we use for quantification. The last part that we come to is, how do we test for antibiotic sensitivity? That is the one that we have as the most important one nowadays. Antibiotic sensitivity kaise test kar rahe? So please note, before I get on to it, there is something known as minimum inhibitory concentration and this is bactericidal. तो दोनों का same word है. That is why I want to emphasize minimum inhibitory concentration means which is the minimum concentration of the antibiotic. Minimum concentration of the antibiotic at which the growth can be inhibited. Bas, kill nahi karna. You don't have to kill the bacteria. You just have to stop its growth. The growth can be inhibited. That is minimum inhibitory concentration, right? On the other hand, what is MBC? MBC says minimum bactericidal concentration. Bactericidal means the minimum concentration needed to kill the bacteria. So you're seeing the difference now. In minimum inhibitory concentration, you were finding out what are you doing to inhibit the bacteria, and bactericidal mein what are you doing to kill the bacteria. That is what is antibiotic sensitivity testing. And now let me tell you that there are many methods by which you do it, which I'm going to explain one by one. Dilution method, disc diffusion method, E-test, एक और strokes test भी होता है, and automated test. सारा कुछ करते हैं. Before that, let me tell you, because we are living in the era of automation, what is that very famous machine in which it is used for automated antibiotic sensitivity testing? So automated antibiotic sensitivity testing is by this white color ka dabba machine has Vitec written on it. That's the name of the machine. So Vitec is done for sensitivity testing. But to reach there, I first have to know what exactly are we doing. So what am I going to start with? I'm starting with the most important method, that is the dilution method. Actually, no one does it nowadays, but still we believe in microbiology, the primitive methods are always the best. So what is the dilution method? So you will say, ma'am, before doing the antibiotic add karne se pehle, at least prepare your patient sample, at least prepare the inoculum. So how do you prepare an inoculum? So that how do you know that, okay, this much of inoculum is good enough? So what are you going to do? You prepare an inoculum. What do you take? You add E. coli to sterile peptone water, right? You are going to take a peptone water, you add E. coli to it, you incubate. So now you will say you are preparing, matlab, what am I trying to do? You will say, ma'am, how do you know E. coli? Abhi patient ka sample nahi liya. You have not taken patient sample. You are preparing your own test in the lab. You take E. coli in peptone water, let E. coli grow. Use grow karne do. After it has grown, you will measure how has the peptone water changed. So initially, the peptone water was just like any other water, transparent like any water. Now when E. coli has grown in it, don't you think it will become turbid? something has grown, it will become turbid. So how will you measure the turbidity? By a spectrophotometer, and that measurements, you will say, ma'am, how do you measure turbidity? The unit is known as the McFarland's standard media. So they say that when you have a 0.5 McFarland's, that means 1.5 into 10 to the power 8 colony forming units have grown. That is your standard. That is a standard inoculum, that whenever. So what is the purpose of this? What do I mean by 0.5 McFarland's prepare kara hai? 0.5 McFarland's means that there are 1.5 into 10 to the power 8 colony forming units per ml, means that this is your standard. See, now if I give you a urinary tract infection ka sample, urine sample, ya fir koi blood sample ya sputum sample, how will you know that what is the right quantity that needs to be tested with antibiotics? You will actually first make it into equivalent to this. You will say, ma'am, I have one standard in my lab. That standard is known as 0.5 McFarland's. If I can make my patient sample equivalent to that, then means I can test it with different antibiotics. So like we say na, that everything has a baseline. Why do you give like grand tests? Because you feel that a grand test will help me assess that this is my baseline. So you say, a previous grand test I got, say for example, 10 marks, next grand test I should get 12 marks. So you are taking a standard कि अच्छा इतना तो होना ही होना है. Same way, I have made a standard in my lab that whatever sample I will get, इतना तो turbidity I should be able to maintain of it. That is 0.5 McFarland's, which corresponds to 1.5 into 10 to the power 8 colony forming units. Now what will I do? ये standard बन गया. Whenever I will get a patient sample, I will make it as much as this and I will compare it. What are the two different dilution methods that we have? There is a micro broth method and there is a macro broth method. Micro broth method mein you can see I'm doing it in a chota wala ELISA plate. And how many wells does ELISA plate have? Come on, so many times I've told you it has 8 over here and it has 12 over here. So totally how many does it have? 96 wells at a time. So many antibiotics I can test. It will save on so much of time and resources. But there is a macro broth method which I do in test tubes, right? And that of course alag test tube mein different dilutions of antibodies will be added, time lagega. So have a look at this diagram. What have they done over here? So abhi ke liye don't do cheating nahi karo, don't read anything that is written, but everyone look at this procedure with me first. So what is this? I have taken a set of test tubes, okay. In that test tube, what have I done? I have taken that standard inoculum is added to each of the test tube. जो भी standard inoculum बनाया था patient का, वो urine का, blood का, जो भी sample था, you have taken that 1 ml standard के सब test tubes में से I will add 1 ml from my patient standard inoculum. But ये जो control है, control is always going to go खाली. Here you are not going to add any antibiotic. बाकी सब में you will start adding antibiotics. And can you see how have I added antibiotics? I have added antibiotics in different concentrations. 0.5 microgram, 1, double कर दिया, 2, 4, 8, 16, 32, 64. And I realized that I have to find out at what concentration will the antibiotic stop growing the bacteria. So I realized कि 0.5 microgram पर तो antibiotic पर तो bacteria grow कर रहा था, 1 microgram पर भी bacteria grow कर रहा था, 2 microgram पर भी bacteria grow कर रहा था. Suddenly, from 4 micrograms and onwards, I realized that there is no bacterial growth seen. I don't see any bacterial growth. So what is the minimum inhibitory concentration for this case? 4 microgram antibiotic minimum I need. If I use 2 microgram of antibiotic, bacteria will continue to grow. But if I use 4 microgram of antibiotic, the bacteria will not grow. Now you can read exactly how it is written in your textbooks. So please remember, the 4 microgram bacterial growth is inhibited. So 4 microgram is going to become the minimum inhibitory concentration. The same procedure has been standardized in all your textbooks like Anant Narayan, in all your CDC guidelines. I've got you the same thing over here so that you know exactly kya aayega. This has told you the very famous minimum inhibitory concentration. So what was this, guys? This was the first method that I wanted to teach you. This was the dilution method. You could either do it in an ELISA plate and you've got your MIC also. Now we are going to go ahead and talk about something called disc diffusion method. So what is a disc diffusion method? It has a better name called Kirby-Boyer disc diffusion method. Kirby-Boyer disc diffusion method. I think you know a couple of things about it. Firstly, you will say, I can see a culture plate, ma'am. Bilkul, dikh raha hai. What is the agar that I have used in this culture plate? It is CAMHA. What is CAMHA? CAMHA means it is cation adjusted Mueller-Hinton-Agar. It is cation adjusted Mueller-Hinton-Agar. Very, very, very famous question of your previous year, that what is the culture media that you use for the Kirby-Boyer disk diffusion method? It is CAMHA. Okay, so पहले agar लिया. After that you say that ma'am I have to put the patient sample. Sample क्या करें? Streak करें या फिर ऐसे हम stab करें? नहीं. I told you that for antibiotic sensitivity testing, like a carpet or a lawn, you are going to spread the sample. So the sample is spread out in the form of a carpet or a lawn culture, and you will incubate it overnight. उसके बाद क्या करोगे? After that you will add these discs. Every disc is a different antibiotic. This is antibiotic A, antibiotic B, C, D. You will say, ma'am, कहीं भी randomly ऐसा disc लगाना? नहीं. The minimum distance between two discs has to be 25 mm. 25 mm at least gap has to be maintained, because at least allow the antibiotic to diffuse and work. Na? Don't say stick them back to back. Allow them to work. So now you're going to see how will you interpret. तो क्या क्या करा? पहले तो you had to maintain an agar. Which agar? Question 1, cation adjusted Mueller-Hinton agar. Question 2, how have you added the sample? You have added the sample by a lawn or a carpet culture. Question 3, once you place the antibiotic discs, what should be the distance? 25 mm. तो अब interpretation पे आते हैं. So let me tell you, if this antibiotic over here means, let us assume bacteria is sensitive. Let me assume bacteria is sensitive. Means, will this antibiotic kill the bacteria? Yes. Can you see? It has killed the bacteria up till here. It has definitely killed the bacteria. So if you see a zone of inhibition, bacteria could not grow in its surrounding. Bacteria is very scared of this antibiotic. It says, "Arey, yahan to I cannot grow only." So if you see a zone of inhibition, this means it's sensitive. So is this antibiotic sensitive? Yes, I can see a zone of inhibition. This antibiotic sensitive? Yes, zone of inhibition. Tell me, is this antibiotic sensitive? Is the bacteria sensitive to this antibiotic? No, ma'am, I don't see any clearing. Bacteria to grow kar raha hai. So I say that whenever there is a zone of inhibition that is seen around each disc, this means the zone of inhibition agar hai, this means there is sensitivity to that antibiotic. Some people say that, you can even measure the diameter of this zone and find out kitna sensitive or not, but that is not going to give you any quantification. So can you find out sensitive है या नहीं है? पता लग गया? Yes. If you saw the zone of inhibition, मतलब sensitive है. But could you calculate the minimum inhibitory concentration? Can you look at this and tell के how much of antibiotic is needed? 4 microgram, 6 microgram? वो नहीं पता लगा मैम. ये problem हो गया. The dilution method was telling me exactly 4 microgram is needed or how much is needed. This told me sensitive or not. It has solved half the purpose, but it has not told me about the quantification. तो क्या करें आप? पहले तो everyone has understood that if zone of inhibition is present, it is sensitive. If zone of inhibition is absent, it is resistant. मतलब अगर खाली खाली एरिया दिखा तो मतलब सेंसिटिव है, नहीं दिखा तो मतलब इट इज resistant. But disadvantage हो गया, it is not telling me the MIC. Okay, what can I do? Let me look at the next method. There is another thing known as the Stokes disk diffusion method. मैम ये भी वही है, इसमें भी कुछ पता नहीं लगा. Only advantage is here I have put an antibiotic. Okay, so for example, whatever antibiotic you are interested in, वो antibiotic लगाया, the same cation adjusted Mueller-Hinton agar, the same carpet or lawn culture, the same antibiotics have been put. सब कुछ same है. Different क्या है? I have kept a control also in the center. I've kept a control strain so that I can compare. Actually, that previous method was not telling me any comparison. Control always tells me the validity of the test, which tells me that हाँ सब कुछ ठीक तो चल रहा है, right? So control was not there over there. यहाँ पर I am comparing. So I saw that the control organism is actually sensitive. I can see a zone of inhibition. Control organism is sensitive, so I can have a comparison. I can basically have a comparison out here. So I could see that now if I have a zone of inhibition around this for the test strain, I have something to compare it with. हाँ ठीक चल रहा है. So basic concept is everything about the Stokes disk diffusion method is the same, but the comparison is going to be provided by the control strain, right? So I have something to compare it with, but the concept is the same. I still don't have an MIC given to me, for which I will do the epsilometer test. What is the epsilometer test? Also known as the E-test. Because now the same story is there. I have again used the cation adjusted Mueller-Hinton agar. I have again used the lawn or the carpet culture media. But now instead of using ऐसा वो round वाला antibiotic discs, I have used a strip. How many? 1, 2, 3, 4, 5, 6. 6 could be adjusted. So 6 antibiotics I have added. If I zoom into this strip, any of them, एक तो you will see इनके ऊपर E लिखा है. So that will anyway tell me it's an epsilometer वाला test. So remember the E tells me the same, but I have this strip is not having the full strip doesn't have this antibiotic in one go. It has different concentrations of the antibiotic. So yes, I am seeing a zone of inhibition. दिख रहा है zone of inhibition. I can see the zone of inhibition. तो मतलब sensitive तो है, but I can also see that where did the zone of inhibition start from? What is the MMIC? यहाँ से शुरू हुआ. मतलब this is the minimum inhibitory concentration. So that is the advantage. Epsilometer test over a Kirby-Boyer or a Stokes disc diffusion test is that this is going to help in providing me the minimal inhibitory concentration. So if I come back to the different methodologies that I told you, number one was that dilution wala, which was telling me MIC. Disc diffusion में Stokes and Kirby-Boyer तो did not, but E-test has definitely told me the MIC. And finally, automated methods, for which I told you a machine called Vitek. Yes, on that note, your culture media chapter comes to an end and you can quickly solve some questions to feel a little more confident. Not true about agars. Is it solidifying around 40 and melting at 95, 98? I told you almost double ka difference hai, theek. Does it have polysaccharide? Correct. Is it a source of nutrition? No. So that is the false statement. It is not a source of nutrition. Coming to the next. A patient suffering from cough and sore throat, fever and chills visited the morning session of the OPD. His sample was sent for culture. The lab technician performed the culture as the figure shown below. कौन सा वाला culture हुआ है? You can see this streaking done. So what kind of a culture is it? It is a streak culture. Next. A patient with complaints of burning sensation when urinating and lower abdominal pain complained with outpatient department with all these complaints. He was on antibiotics for a few days, but there was no improvement. Urine sample was collected and sent for antibiotic sensitivity testing. Which of the most commonly disc diffusion method that is performed? So what is the most common one that is done? अच्छा, अब सारे ही वो हैं, but the most common one that is done is actually the Kirby-Boyer disc diffusion method as per MCQs. But up to we have become very, very advanced. But yeah, as per MCQs, Kirby-Boyer disc diffusion method. Let's move on to the next question. There is a 35-year-old male resident presented with fever and cough, and he was well until 3 days earlier when he suffered the onset of nasal stuffiness, mild sore throat and a cough productive of small amounts of clear sputum. The physician sent him for a sputum culture and the microbiologist used a blood agar. What type of a media is a blood agar? Now I know blood agar में blood है तो कुछ तो rich बनाया. It is definitely an enriched media, but apart from it being an enriched media, do you also remember that it was something to do with the difference? Yes, was it telling me hemolysis? So it is also a differential media that you need to consider over here. Let's pick up one more question. Look at this. A 40-year-old female presented to the OPD with burning sensation and urination, pelvic pain, increase in urge frequency while urination, classical urinary tract infection की बात हो रही है. Antibiotics given showed no results. So the sample was collected, sent for an antibiotic sensitivity testing. Last line and retrospective helps us. The inoculum prepared should match which of the following McFarland's standard. So if you remember, I'll take you back. 0.5 वाला कुछ याद आ रहा है? 0.5 McFarland's, which is equal to 1.5 into 10 to the power 8 colony forming units. That is going to be your standard, and here you have McFarland's standard 0.5 written as your answer. Simple. Coming to the next. There is a 22-year-old female who presents with, again, same story of that female going on and how they are changing the question. UTI chale ja raha hai, frequency, urgency, painful micturition, you're suspecting UTI, urine has been sent to the microbiologist. Microbiologist decides to do culture on McConkey agar. Which of the following is there in McConkey? I know McConkey has plant. मतलब peptone, options में नहीं दिया. Lactose, दिया है. Agar, not in the options. Neutral red, there in the options. Taurocolate. Sodium taurocolate is a bile salt. So what is your answer? Your answer is all of the above. The last question that we have, Antibiotic sensitivity testing is carried out to determine the appropriate antibiotics to be used for a particular strain isolated from clinical specimens. The most commonly used medium for antibiotic sensitivity testing was CAMHA. CAMHA is what? Cation adjusted Mueller-Hinton agar. So the answer over here becomes option C. And on that note, we have wrapped up our culture media. Still, if you have, if you have not studied bacteriology, this must have been such an overabundance of knowledge for you. If you've studied bacteriology, you might be seeing this on a 2x speed because it must have been a repetition of a lot of things. Thank you.